incomplete dominance. This best, easiest one to look at is snapdragon flowers. Dominant is red. Recessive is white. So if we do a Punnett square, with heterozygous parents, or I mean with, let's make it homozygous parents. Let's make the mother flower, female flower, homozygous for white, and the male homozygous for red. This means that all of the offspring would be heterozygous. This also means that the color of each offspring would be pink. Red is not totally dominant. So, incomplete dominance, the dominant isn't completely dominant, and so it's a actual mixture of the two. Codominance. This means shared pair dominance. Let's use flowers again. You have the flower, the dominant, one of them dominant is red. And then we have another dominant one is white. Now there could be recessive in there, but let's just make a Punnett square with two homozygous of the dominants here. So let's make the mom homozygous, or the female flower homozygous red, and let's make the male homozygous white. This means we are going to have, and now you're seeing I'm using two dominants because two letters, not normal for a genotype, but in this place because they're both dominant. Now, this would mean that all the offspring are going to be red, and white. I'm writing fairly small, but you can see. So there would basically be patches of red and patches of white because they are codominant. Now, codominance in humans. We like to use blood types. Now, we have type A, type B, type AB, and type O. Now, what it is that we have this red blood cell, and if it's type A, it'll have what we call an antigen. It's a protein on here that if you do not have the protein, if you don't have it, it will cause you to make an antibody against it, but it's in your blood, so you don't. So that's type A. We call this immunoglobulin A. And type A could be homozygous, or it could be what we call heterozygous with the recessive. The recessive is type O, and type O can only be 2 of those. They don't make any of this. Type B, you would have a different antigen, different protein on the surface of the cellular membrane. Type B would have the immunoglobulin B. That stands for a protein that has something to do with immunity. It could be either homozygous or heterozygous. Now, type AB would have both of these proteins on there. And the proteins are on the plasma membrane. Just like the flower that had red patches and white patches, these would have both of these proteins on there, and that is type AB. So type AB would always be this IA, immunoglobulin A, and immunoglobulin B. Immune system globulin, a globular protein. So you have 4 genotypes, and you have 6 genotypes and 4 phenotypes. Now, of course, for type O, the red blood cell would have nothing out there. None of these proteins. So doing a Punnett square, let's do one Punnett square here. And let's take the mother is type O, and the father is AB. That means we would have 50-50 chance of getting a child with type A and a 50-50 chance of getting a child with type B. Let's do another one. let's say we know the mother is O, and we know the father is A, but we do not know if they are homozygous or heterozygous. Well, that means we'd have, for sure, we know what's going to be in here, but here is what's going to determine whether or not the father is homozygous. Basically, if we get a child that is O, we know the father is homozygous, heterozygous. If we get all children that are A, we will assume that the father is homozygous. However, it could happen that you have 4 kids and all of them are A, but you're still heterozygous, because realize that when you talk about probability, you're usually thinking of large numbers. Well, 4 children is not a large number, meaning, but as the more children they have, the less likely you're going to have all type A children if the father is heterozygous. But we know for certain that if we get a child that is O, that the father is heterozygous. Now, why do we worry about the about blood types? If what happens if someone who's type B gets type A blood, their immune system doesn't recognize this protein on the blood. It starts building antibodies against it. Now, if they've never had type A blood before, they might be fine, they might survive, because by the time their immune system has built enough antibodies to attack this blood might already be out, meaning blood cells don't last forever, and antibodies take time to make. But if the person had already been exposed to type A blood, they would build antibodies very quickly. They would already have the antibodies in the blood, and it would clump up here and the person would die. Now, does that mean you can always have blood, that it's okay the first time? Well, no, because during pregnancy, the mother's blood might be a different type, and if it leaks into, and so the child becomes, the child's immune system identifies it where it gets in there, so it starts building antibodies. Well, then you're already you could not have that blood, so you play it safe in that.